Thank you, Matt. And I've been having great time with the meeting. I hope everybody can see my presentation.
Looks good.
Okay, we are fashionably 5 minutes late, so I hope I'll get 5 minutes, or maybe 7 minutes. So we'll get 7 minutes more at the. I'll try to make up some time. So what I want to do today is to try to present metabolomics tools and resources for the research community that we have developed over 10 plus years. So this is a 10 plus years of work. I want to summarize in the next 30 minutes and tell you how it is for the community. So when I started thinking about this presentation, I asked myself, what would I like to do? And I told myself, I need to really do it as a community member. And when I talk about that, I want to tell you it's a resource for the larger community. It is not a resource for such people who know they can do everything on their own. So we don't need to worry about it. I mean, I know Oliver can take care of everything. It's meant for people like this who really worry about, Workbench and the physicians and clinicians and researchers, biomedical researchers. And this is what the entire scope of what we wanted to do in the beginning was. So towards this, we asked the question, what should the metabolomics resource provide? Number one, it should have the ability to store data and metadata. And this is really for data generators. And we want to make it very easy for data generators, even a graduate student who is generating data in the lab, to be able to really, to store data very easily. It should be possible to identify studies that are available. I mean, all studies that are available in a one-stop shop environment. And this is findability. And I'll convince you today that we are really findable very easily. Ability to query and search and obtain such data. I mean, really, one should be able to query such data. This is people underplay this notion of querying and searching. I mean, if you have to write SQL queries, if a person has to learn how to write database queries to query a data, then it is not useful for a biologist. And so that's really one of the key issues that comes up. Ability to analyze on the fly with minimal computational expertise. You want to be able to say that, hey, I have my pet lipid or my pet amino acid, and I want to really look at what other studies have found this to be significant in their, quantitatively significant in their, research. And so these types of questions are common. I'm going to try to look through the lens of such a person and answer those questions today for you. Ability to integrate data from different studies, tools and interoperability. In fact, this is critical because we want to compare, for example, two different cancer studies. You want to ask the question, what's common, what's similar? Ability to visualize results from analysis in biologically meaningful formats. And this is often the largest barrier for people like me and people like Owen and others who design these things, because finding GUIs which are really aesthetically pleasing, usable, and meaningful is always a challenge for all of us. Ability to download data and results in formats that can be biologically meaningful through easy interfaces. These are, again, user interface questions. And these are the challenges that anybody who's designing an infrastructure faces in order to be facile for the community. I'm going to wear the lens of a biomedical researcher today and walk you through what we have developed over the last 10 years. So this is, all of you know, this is the NMDR Metabolomics Workbench. As of yesterday, there were 2,099 studies that have been processed by NMDR. 1,829 are publicly available studies, and reminder will be made available when the embargo status is lifted. And this is as of yesterday. So you can see that we really continue to grow dramatically, rapidly. I think Owen mentioned the similar number yesterday. It was a few less. And so this is really where we are. Now I want to walk you through what do I look like. So if I'm a submitter, if I'm a person, graduate student in Rick Yates's lab generating data, and I want to submit this, then it's very, very easy to submit. I'm going to walk you through this in a minute. You can create what's called the MWTab file. Very easy to create. It contains very easy formats like metadata, project, study, etc., data, and then you can upload the data. This is really fairly straightforward. Many of you have experienced it, and Owen provides a beautiful tutorial for you to go through this and be able to upload data. This is a data repository. We allow browsing and searching, statistical analysis, downloading targeted measurements, downloading MWTab files at various levels, and so forth. I'll walk you through this in a minute. Imagine I wanted to access and download a data. I'm an end user. I want to download a data that contains NAD plus in neurodegeneration. This is the University of Florida study on humans. And this data was uploaded in 2018. Imagine I want to download it. All I need to do is I can go in and click on that button. I get this information with reference to what the study is about, a summary version. And then I can go on to look at perform. I do any of these options I have. For example, I want to download named metabolites. I can go in and download named metabolites, download MW tab file as a whole, and I can look at all the statistical analysis in a very straightforward, easy manner. I don't need to know deep programming of any kind. How can I access it for the searching and browsing data? And how can I access this data? I can do it by using REST service that Owens developed. And this REST service is very straightforward. It gives you Metabolomics REST service provides you with structure-specific information, metadata information, gene, protein, pathway-specific information, and mass spec search-specific information. So you can get all of this in very easy formats. What about untargeted mass spectrometry, which is really becoming very common these days? We have a lot of untargeted mass spec files. So how do we deal with untargeted mass spec files? So here's what we do. The strategy for downloading it is strategy for us is to curate it, filter, normalize, and select conditions. We identify from the untargeted case what are the significant features. You can view it in a Volcano plot. You can search other MS studies containing named metabolites by M over Z value. You can search metabolite databases, MS databases by M over Z value from computationally generated lipids. You can look at coelution with known metabolites to get some clues on what they are. We can generate table of correlations with all identified metabolites, even if you didn't know what the metabolite was. And then to see what is the guilt by association factor and project on mass defect plot and clues to metabolite classification. So this we provide you just on the fly for you to look at. In fact, yesterday, Zhu Shadu presented a beautiful talk on searching for studies in similar mass spectra. We use ADAP KDB that was developed by Zhu and her group, and it allows a person to search for MS data for features, unique pairs of mass and retention time, and then it provides this entire pipeline or workflow of searching for mass spec data from unknowns. And here is how ADAP KDB works. You saw this yesterday in much more excruciating detail, but you can come to the workbench. You can go through ADAP data pre-processing, annotated mass spectra, look at hierarchical clustering, construct consensus spectra, and then you can go to the ADAP KDB portal for further analysis. So this is really very easy for somebody who wants to analyze unknown mass spectrometry, untargeted mass spectrometry. Searching the Metabolomics Workbench for metabolites. Often, this is the question that I get every time people talk to me about Metabolomics Workbench. They ask me, hey, can I find my favorite metabolite? Can I find my favorite metabolite in your large set of studies? Not only you can find your metabolite, you can find quantitative estimates of this metabolite. I'm going to show you that in just a second. So you can look at, we developed, in fact, Owen and others developed a RefMet reference list of metabolite names. And this is a standardized reference. What does it do? It allows you to harmonize data across studies and facilitates integrative analysis. In fact, this is one of the banes of metabolomics. There's so many names. Distinct people use different names. And so how do we harmonize that? So Owen's RefMet provides a unique way of nomenclaturing and organizing the data. And then here's an example. RefMet, for example, all of you know this. Owens presented a number of talks on RefMet. We published this in Nature Methods in 2020. It's a nomenclature for metabolites widely adopted now, and then easily searchable and queryable across the database. So when you want to search, for example, we now provide you with a quantitative way of looking at data of your metabolite. MetStat, which is, imagine that I'm a bench researcher. I measure a given metabolite. I say proline. I want to find out, is my proline measured in my cell or in my tissue? Is it an outlier or is it an average human value? I can ask that question today without going into any programming. I can do meta-analysis on the metabolites across all studies. I can take all the studies. For example, there's an easy user interface. I can pick analysis type. For example, I can choose all or only positive ion mode, certain mass spectrometry. I can do chromatography type, disease type. I can contain names of this, provide a name for this. For example, human studies with blood is what I choose. And then I'm asking the question, find me all the prolines that are available in human studies on blood. Click on this point. This is very easy. And then when you click on it, list of human studies that report proline comes up. And then you can click on a study that you're interested in, asking, is this an outlier? You can even upload your own data. It doesn't have to be a data, or you can look at a glance what the data is. When you look at it, for example, here, this is the average value of proline. If you take one given study, proline is here, which is an outlier. And so you can get study 1354 contains a proline outlier. So it's really easy for you to look at the entire, and as studies grow more and more and more, we are going to get the metabotyping of humans, which is the average profile of all metabolites, many metabolites, important metabolites in human samples. This is human plasma. As the data grows, we will see it in human tissues. And this is one of the things we argued yesterday and day before in terms of having to do metabolomics on human tissues. Here's another example, LPC16 and 0 and creatinine. Creatinine is commonly explored in metabolic panels. And you can see, for example, what is the average value and what is the deviation, what is outlier and so forth. Same thing is true for hypoxanthine or triacylglycerol. You can look at all these values, just quantitative values at a glance, and look at the distribution profile. So this is, in fact, if you want to look at, hey, what metabolites are found across all studies in blood or 100 studies in blood, you can look at it and see this is a distribution of metabolites classes that are present across all these cases. So it's very easy for you to navigate. Art Castle started off the meeting by presenting this, our workflow for Metabolomics Analysis Foundations. Metabolomics Workbench, one of the largest used resource in the Metabolomics Workbench is the tool set. We have provided a whole list of tools. In fact, we provide statistics. I mean, you name it. We have got all the tools: normalization, clustering, univariate analysis, multivariate analysis, mapping metabolites to pathways, comparative studies across analysis across studies, untargeted mass spectrometry studies, NMR experiment studies, volcano plots, etc. So we provide you with a whole list, and this is for an average biologist. It is not meant for Oliver Feen. And so here's an example. I'm going to download, for example, big data solutions. This is a TEDdy study with 11,560 samples. I want to download this MW tab file. I want to look at distribution of metabolites in this study, in this TEDdy study. I can look at it, and I can look at the statistical significance of these values. Imagine you've got, I'm talking about not a small set. I'm talking about 11,560 samples with 1.6 million data points. And we can look at the distribution trivially in terms of what the distribution looks like. And you can dive into more details in terms of depth. So here's an example of, study-specific analysis of the very first study we received, FAD-B induction experiment. You can run analysis of various kinds. You can get volcano plots, correlation plots, PLS-DA scores. You can do all these kinds of analysis. I'll give you one more example as we go along. So here is, for example, multi-condition dot analysis plot. Here is an experiment. I'm asking, in methionine stress, what's the sensitivity of human breast cancer cells? A commonly asked question. And so imagine I want to compare different experiments, control experiments. This is the control experiment. I want to look at the control experiment value. Here is a dot plot of the metabolite changes associated with methionine stress. For example, in early time period, ceramides are upregulated. With increasing time, we have got the triacylglycerols. I can get all of this at a glance without having to go, and I can always dive into deeper quantitative analysis at this point. Here's an example of study 1140. I want to do correlation, clustered correlation analysis, perform statistical analysis. I want to look at the data. The data, here is an example of data where we look at Parkinson's disease in mouse, DJ1 knockout mouse models. And imagine I'm the experimenter doing this work, and I want to compare, I want to compare, for example, genotype with wild types, and this is the control set, and I'm comparing it with group 2, which is the knockout group. And I'm going to compare this. I'm going to do volcano plots. I immediately get what is significant. In an ANOVA experiment, I click on the ANOVA button. I get what is significant. I can then go on to look at a PLS-DA plot. You can clearly see the distinction between these two classes. I can look at all the important metabolites that are differentially scored in my OPLS-DA plot. All of this is just on the fly. I'm doing this. How can the data be integrated to other omics data? We talked yesterday ad nauseum about the future of metabolomics. The future of metabolomics lies in our ability to provide a systems level perspective of cellular function and phenotypes, not just simply metabolome alone. And so how can we integrate? Here's an example from a lipid maps experiments that we did. Here's a RAW macrophage cell treated with lipopolysaccharide and other, with ATP and lipopolysaccharide. And then here's an immediate comparison. You can generate heat maps. You can look at pathway-based integration. And you download the statistical tools that we provide, and you can use it to create on your own. Here's what you get at the overall mechanism. Here's the overall mechanism of how this integration provides you with KLA and ATP. You activate not only the NF-kappa B, but the AP1 pathway, giving rise to icosanoid metabolic changes. So these are the kinds of things you can derive as an end user. How about the data mining in terms of, I'm sorry, I already mentioned this data mining for untargeted data. Here's an example I'm going to give you of searching through changes in plasma lipidome after short and long-term exposure to glucocorticoid exposure. Here's an example. I can look at comparison between two groups, treat with prednisone and treatment group with prednisone sample time point before and after treatment. And so I can look at the Volcano plot, look at significance in terms of hexacylceramides and phosphoethanolamines are increased. This is all immediate analysis that we can perform. Here's to say another experiment, 1140, the same experiment, prednisone. I can generate correlation plots. We had a lot of help from the tools group, and they generated a lot of wonderful tools, and I'm going to show you an example. Here's a correlation plot from using the Metabolomics Workbench tools. Here's a correlation plot. We can do a hierarchical analysis using for all the correlated metabolites. And here's an example of DSPC algorithm from Ala Karnowski and George Michaelides groups, Chuck Burant, their groups. We have incorporated into the Workbench. We can look at comparison groups, look at unbiased correlation here. We can translate that correlation into clustering by groups in two experimental conditions. Yeah, end user can come and say, hey, what's correlated? I can look at named metabolites. You can see immediately phosphatidylcholines or a cluster of correlated metabolites here. And then they can go on to look at, base it on RefMet classification and say that which of them are phosphatidylcholines and which of them are phosphatidylethanolamines, O-glycosylated phosphatidylcholines, etc. And you can parse it. All of these tools are easy to parse on the Metabolomics Workbench. Metabolites for fold change between two conditions. I can look at fold changes. I can look at quantitative values, look at correlation coefficient cutoffs, look at fold change cutoffs. You can vary this as an end user. So here's an example of such an experiment performed with 0.7 cutoff with 1.2 fold change. I'm looking at the University of Michigan test data set, and here's what I get as being significantly regulated. So you can see all of this tools navigation is really easy for an end user. Here's a more detailed version of the D-bias spatial correlation, sparse partial correlation algorithm. When we incorporate it, we can look at the acylation involving arachidonic acid. We show acylation involving oleic acid, and same chain decomposition involving phosphatidylinositol 36:3 and into 38:3. So you can look at all of this in fairly routine ways. So MD Anderson group, yesterday we had a beautiful presentation from John on Metabolomics browser and next generation clustered heat map. I'm sure he showed you all the details. I want to just bring it up because this is an extremely useful tool for a bench biologist to go in and look at and navigate and expand and look at a specific region. And John showed this yesterday very nicely. We have it on the Workbench. You can, for example, I took the study 1386 and I did this really trivially. And then we look at this, example, we can look at choose that study in the Metabolomics browser. And then once I choose it, I get the clustering, PCA plot. I can go and do a heat map. I can go and zoom in a specific region and pick up heat map detail and then look at how do phosphatidylethanolamines and phosphatidylserines change. And you can clearly see that there's a significant difference in between these two conditions. So really, we are able to navigate this in a fairly easy way. All of this is possible because of the 10 years of work that's gone into Metabolomics Workbench as a resource for the community. When we sign off this 10 years, I want all of us to understand that this is not just simply a database. We created an infrastructure for the community to use. I hope the infrastructure will sustain and keep going so that the community can really benefit from the use of this. We recently developed something. We asked the question, how do you go from I mean, all the jazz today is in integrating genomic data, gene expression data, metabolomic data. How do we link gene data to metabolite data? This is in collaboration with the CFD Metabolomics group and funded by CFDE. We essentially created a way to go from a gene to genes to pathways to reactions to metabolites to studies in a seamless way. If you're an end user, all you have to do is to go in and choose the gene of interest, find all metabolites which are either biosynthesized or metabolized or associated with that gene. You get the reactions, you get the pathways, you get the studies. I'll give you an example. For example, I want to choose PNPLA3 gene involved in fatty liver disease in humans, in blood, in fatty liver disease. I want to choose these things, and then I want to look at what are the pathways, reactions, metabolite studies involved in this. I can go and look at PNPLA3. I can look at the ensemble ID, or in fact, Mano developed a way of navigating between multiple IDs. You can give any ID that you want. It takes you to any genomic database and gets you the equivalence ID. And then you can look at pathways, you can look at reactions, you can look at, for example, here DAG is hydrolyzed by 3-DAG is hydrolyzed to 2-MAG by PNPLA 2 and 3. This is the pathway, this is the reaction. You can get details of the reaction and so forth. So we provide you really with a way of looking at in great detail from genes to proteins to metabolites. So here's an example of a link to RefMet. We can look at the triacylglycerol that came up in PNPLA3. Ask the question, hey, find out what the reactions are, find out the link to metabolite statistics, what is available today in the Metabolomics Workbench in blood, and we can look at that, and you can look at the distribution. In our group, we developed also a MET-ENP, which is Metabolite Enrichment and Pathways, a one-stop shop. Imagine you're an end user doing mass spectrometry in your laboratory. You get a bunch of metabolites, targeted metabolites, and you want to go in and ask, what's the function? What is a functional enrichment? Just like in gene ontology, you can look at functional enrichment. It allows users to map metabolites to standardized metabolite classes, calculate significantly altered metabolites, calculates enrichment score of metabolite classes, maps to pathways and species of choice, calculates enrichment score for pathways, get the gene reaction information, supports various visualization plots. All of this from the Metabolomics Workbench. And here you can choose a study. Imagine this study chooses the canine plasma lipidome after short-term, long-term excess treatment with glucocorticoids. And here is exposure. This is in dogs. And we are looking at the data set with 3 analytical methods: reverse phase MS, LCMS HELIC, and normal stationary phase MS. And then here's what we do. We look at the treatment group. We look at combined factors, 0 days, 4 days, 0 weeks, and 25 weeks. And here is, you can do comparisons across. This is all the user interface where a user can choose very easily what options they want. And then here's what the plots look like. You can click on any one of these. And here is a volcano plot. It tells you, for example, phosphatidylcholines are significantly upregulated across the 2 conditions you're comparing. And the triacylglycerols and ceramides are downregulated. Here is the up and downregulated distribution plot that you can look at. You can look at pathways, the same thing. You can look at the enrichment plot. You can look at all the pathways. Here is a pathways involving the arachidonic acid metabolism. Here's a pathway involving the sphingomyelin metabolism, sphingolipid metabolism. And so you can look at all these things at a glance, and you can click on it and go and look at individual details. So this is what MET-ENP does. So all of this is available on the Metabolomics Workbench. With Katerina Ketris's group and Ghosh's group, we are able to import tools from Colorado group. MSCAT provides you with an ability to workflow. In fact, most people who work in metabolomics really want a workflow to go from A to Z. So we can dynamically develop a workflow here. MSCAT allows you to do that. I think Katerina is going to mention that, and there is a mention of that in the previous talks. And so this is something we developed. MSCAT allows you to provide you with a software catalog. Again, to the community, this is extremely useful. And tools from the Colorado group, we have many other tools. We have Docker tools. We created Shiny App tools, and the Dockerized and Shiny App tools are available on the cloud. And talking of cloud, before I go to cloud, we have all of these tools available on, you can do it on iPad, iPhone. We can look at it in mobile phone apps. We make it easy for an end user to work at. Young people today, Old people like me don't really go to, do these things on mobile iPhones, but young people today fly on planes and do these things on iPhones. And so this is really a capability that Owen and others have developed. And so cloud applications, we provide cloud applications and resources on the Metabolomics Workbench. And when the sun sets on the Metabolomics Workbench, it will still be cloud compatible, cloud available. So there'll be all the tools, web tools, Docker tools, Shiny apps, and everything is available. These are all the tools that's currently available on the Workbench. We can perform every analysis through MyBinder, Python versions, and MWR scripts. We are hoping to develop some of these on app eaters, like MetNP was developed on Avi Mayan's app eater. And so we are going to use these Jupyter notebooks and app eaters to provide easy to use tools for the community. So these tools are really, versatile to be used on the cloud. So the Workbench Cloud is based on the National Data Services Labs Workbench. This was started in 2016, thanks to our collaborators in the San Diego Supercomputer Center, who are part of the Metabolomics Workbench. So we are able to use these tools to, they helped us in terms of building this Workbench Cloud. The Workbench Cloud allows you to navigate in a very easy touch format all the tools that are available. So here's Jupyter notebooks that are available. We can launch these Jupyter notebooks from the web browser via MyBinder. This is available also on GitHub. So people can download these, modify the Jupyter notebooks, edit them, and use them as they want. What can you do? You can retrieve, for example, study data, compound data, RefMed data, etc. You can do multivariate analysis. You can do clustering and correlation, data normalization, as you please. So collaborations with other centers, we have developed over a year's number of collaborations. I only list 4. We have many, many more. We have TopMed, we have Precision Nutrition, we have all kinds of collaborations. To give you some examples, we have collaboration with Cheer Data Center. In fact, we helped them to launch their initial website with reference to metabolomics. Lipid Maps is started in our laboratory. Owen's been still integrally involved in Lipid Maps jointly with Cardiff and by Abraham Institute. MotorPack, we have strong collaboration with MotorPack. LionWeb, Lipid Ontology, we are dynamically working with them. We have collaboration with the European Bioinformatics Group. In fact, we provide Metabolomics Change, which is a joint operation with the European EBI Group. And the EBI Group and us are jointly developed with Metabolites and Metabolomics Repository in France and in Metabolomics Workbench. It's a one-stop shop. You can go into a Metabolomics Change and find all the studies that are present and then go and navigate the studies in the respective places. So this is kind of a very rapid half-hour tour of the Workbench. And I put myself into the head of a bench biology user. And I hope I've convinced you that this is really a resource that the community can use in a seamless manner. Are there still things for us to do? No question about it. We have things to do still. What is missing? What is missing is really, we want to maintain these interfaces. We want to increase the number of studies. We have 2,000 plus studies containing, as Owen pointed out yesterday, millions of metabolites. We want to increase this. We want to make this into a one-stop shop resource. As Owen pointed out, now 50% of the studies are coming from outside the country. And the usage is also similar. In fact, when I go to other countries in Europe or in Asia, I always get the comment, hey, I didn't know that, I didn't realize how useful Metabolomics Workbench was. And so this is really a common thing. We get calls from many companies in these countries saying, hey, can we, use your Metabolomics Workbench as a commercial tool? And so these are questions that are commonly posed to us. So I want to convince you that this is absolutely remarkable that we have this. And I want to, last point I want to mention to you is that this Workbench is really not, it's a contribution from a number of people. Owen's been instrumental in this Workbench. I want to really point out, he's really been a pioneer in this area. Mano has been working significantly in all the algorithms and methods. Sumana and Neela are curators now, as Owen pointed out. Manish used to work in the group, developed a number of tools, and especially chemistry-based tools. Kenan used to be one of the key curators for Metabolomics Workbench. Shakti works on tools. Kim was instrumental in curation, and she was involved also in helping with METNP. Srini is absolutely instrumental in working with human studies. Lena is one of the key curators for us today. And Kevin and Christine are the ones involved with all the cloud-based computing and environment. And of course, we would not have done this without all the cores the data DTCs. The first 5 years, we had excellent collaboration with the regional centers. The second 5 years, we had the data course, the lipid course, the metabolome course and so forth. And so they helped us immensely. And a shout out goes to Rick and Mike for their strong, support in terms of collaboration. So this is what made our Workbench and NMDR possible for the community. I hope I've convinced you that this is something which is a unique and immense resource for the community. Please ask me any questions, make any suggestions. We would love to work with you closely in terms of the next generation Workbench and where we want to go. Thank you for your attention. And I'm just 1 minute over, I guess. Thank you.
Excellent. Thank you, Shankar. I think we can take time for at least one question, if it's relatively short.
Either everybody is convinced it's a good resource or people didn't understand what I was saying. No, I'm just joking.
Yeah, I counted to 8. It takes 8 seconds of silence for an introvert to speak. So we didn't get any introverts off the sidelines. So we'll move on to.
How about an extrovert? How about
okay.
an extrovert then?
All right.
I want to say that is a breathtaking summary of a fantastic resource. And Shankar, Owen et al., we've very much appreciated working with you to incorporate our tools into the armamentarium that you've provided. I must admit that I took screenshots of every slide that you presented, and we'll be going over it in our working groups over the next weeks. Thank you.
John, thank you very much. And I will make this presentation available on the website. You've seen much of this, many of you have seen much of this before in different forms, but I try to integrate all of this together for the community at large, especially because this is the 5-year report at the end of the funding period, 10-year report end of the funding period. So I thought it'll be good for us to summarize. And I don't know if NIH folks are around, but if they are, this is really thanks to you. I forgot to really thank NIH, for really helping us build this resource. And unlike other program officers or programs in NIH, I have to say that I've had so much input from the program. This is really, it's been a privilege to work with all the program people who have been involved in this. And this is not a light statement or a flattering statement. I sincerely mean it. The number of calls and input Art Castle, Padma, and others have provided me is really immense. And with the CFDE now with George and Christy and others, it's really amazing amount of input into what we can improve, how we can improve the resource. So thank you all.
Do you give any sort of online training to the group or to any institutions if they get in touch with you?
Absolutely. Owen's done training in many different forums, and Owen provides online. There's a training tool, and we will be happy to work with you in terms of helping provide online training for any of these kinds of things. It'll be a pleasure for us to do that.
Thank you.
Okay, I think we should move on.